Hello. Welcome to the Problem Set 6 worksheet video. Let me share my screen with you. And let's see if we can get started. This topic is variation in chromosome number and structure. So first, we're going to start off with some vocabulary. Aneuploidy means that the chromosome number in a typical somatic cell from that organism is not an exact multiple of the haploid number. So something like we have an extra chromosome or we're missing one or two chromosomes. Polyploidy occurs when we have extra sets of chromosomes. So we think of haploid as one of every chromosome, diploid as two, triploid, tetraploid, and so forth. So let's look at this problem. Corn, which is identified as Zea mays, has 20 chromosomes in a typical somatic cell. It's described as 2n equals 20. So we want to say we have a corn plant that's monosomic, or whatever that description is. We want to tell how many chromosomes there are in an interphase somatic cell. So for a monosomic corn plant, monosomy means we're missing one chromosome. The normal corn plant is 2n equals 20. So 20 minus 1, a monosomic plant only has 19 chromosomes. A trisomic plant has an extra chromosome. And you may think about like trisomy 21 in people, which causes Down syndrome. So in corn, a trisomic corn plant would have 20 plus 1, or 21 chromosomes. A haploid corn plant would only have one of every chromosome type. So half of 20, 10 chromosomes. Diploid is exactly what corn is, 2n equals 20. Those have 20 chromosomes. A double monosomic plant means that it's missing two chromosomes, and those two chromosomes are from different pair numbers. So 20 minus 2 is 18. A nullosomic plant will also have 18 chromosomes. It's missing two chromosomes, but this time the two chromosomes are both the same pair number, so it doesn't have any copies of that pair number. I encourage you to go back to the lecture and practice with the vocabulary in the lecture as well. One of the big topics for this part of the unit was chromosome aberrations. And we talked about various types, the four main types being duplications, deletions, inversions, and translocations. But we talked about various subtypes of each of those. And I encourage you to go back to the lectures and make sure that you are very clear on the vocabulary of those. Now let's look at this one. It says an individual has it says 4 chromosomes. I think there are only 2 chromosomes there. So it has the 2 chromosomes shown below. The periods represent the centromere. So the letters are the genes on the chromosomes, and the period is the centromere. We want to figure out what type of aberration is present. Well, since there are only 2 chromosomes, we know it's not going to be a reciprocal translocation, or even a non-reciprocal or a Robertsonian translocation. So that leaves duplications, deletions, and inversions. If we look at both of these lines, all of the letters are here, and they're here in one copy in both of those chromosomes. So we do not have a duplication where we would have 2 copies of the same letter, and we don't have a deletion where one chromosome would have a letter and the other would be missing it. So let's see if we can look a little bit more closely. If I start at this end, it goes WTS on that one and WTK on this one. So the first part's the same, and then it gets different. Let's start from the other end. It goes ADG ups, either K or S. So the part at the very end, again, is the same, but then this part is different. Look closely at this. If I read the left one from left to right and the right one from right to left, they read the same. S, R, P, M, centromere K. That means that we've inverted that segment. We don't know which is the original, and we don't know which is the inverted, but we see that there is an inversion. Notice that the centromere is among the area where the inversion occurs. That means we have a pericentric inversion. A paracentric inversion would mean that the centromere was outside of the inverted sequence. The next part says, draw the structure that would form after synapsis in this individual. Okay, all inversions, both paracentric and paracentric, have the same general structure. I like to draw this part first. There's one chromatid of the chromosome, and synapsis occurs when there are two chromatids on every chromosome. So here's the second one. We'll put the centromeres in a little bit. So one chromosome curls, and the other one goes in, hops over, and goes out. And it also has two chromatids, so we'll draw that second chromatid. Now, it doesn't matter which of these you decide to label first, because at the end of the day, both of these will be labeled on your diagram. So I like to look at the inverted loop, and I like boxing it off like I've done here, because before the first line comes before the bubble of the inversion loop, and after the last line comes after that loop. So I'll put WT before and GDA after. The other cool thing is that since this is synapsed, directly across on the other chromosome are the same genes. So I can go ahead and label those. The part between those two lines is the part that's on the loop. So I'm just going to label it using this one going from left to right, but I would have been equally okay using that one going left to right. So SRPM centromere K. Now, if I want to check the labels on this, it goes WT, and then going around this way, it will be K centromere M P R S. Didn't quite leave enough room to make that look neat. But if you look at the key online, it will look a little better. So now it says, suppose a crossover occurs between R and P. In order to do this, we have to cross between non-sister chromatids. So don't cross the top two, they're connected to the same centromere. We have to cross one chromosome that's connected to this centromere and one that's connected to that. The easiest way to do that is to cross the two middle ones. So I've drawn the X for the crossover. And then the last part wants us to tell what are the 4 gamete types that can be made from this individual, and also classify those as viable or not viable. So at the end of the day, I'm going to have 4 lines. Each will be tracing one of those 4 lines. And it doesn't matter what order you do them in. You can start at any end and then let it go and take it wherever it goes. So I like to start at the top on the left. So WT, SR, P, M centromere K, GDA. And that took me to the top on the right. So I'm just going to put those in yellow. WT, SR, P, M, K, GDA. And I'll write that on my thing right here. So now I have my chromosome. This is viable. This is one of these 2 that we started with. And if you have this chromosome in the gamete or that chromosome in the gamete, you have exactly 1 copy of every gene and it is viable. All right, let's try another one. I think I'll start with this second line right there. And that has WT SR cross. So I've gone to the lower line. PM centromere K. Oh my, it's pulling me back to TW, the lowest line. Wow. So this one is not going to be viable. And the reason is it has 2 copies of WT and it doesn't have any GDA. So it doesn't have 1 copy of every gene. But let's write it out. Make my line. WTSRP M centromere KTW. So WTSR cross down, but I'm still picking up PM centromere KTW. Okay, well, we'll try another. I haven't done this third line down right here, so we'll start there. That one. I'm doing this one in pink. WT K centromere M PRS, and this is coming to be the third line there, GDA. Let's make a line for this one. Remember, we just said this was inviable. So this one is also viable. It has 1 of every gene, and it actually has that inverted order right there. So we'll write it out. WT K centromere M PRS GD. So both of those are viable. Each one is in a different gamete, both viable. And the last one, let's see, I think I have one more color here. Notice I've used all of these ends. So this last one's going to start on the right and it's going to circle me back around to the right. So I'm going to start with that second end. ADG, go up, K centromere M, P, cross down, R, S, whoops, GDA. So this is the other one that's not viable. A, D, G, K centromere M, P cross down RS, GDA. And it's not viable because there are 2 copies of A, D, and G. So these are each in different gametes. So 1 gamete type, 2 gamete types. Both of these are not viable. 1 gamete type, 2 gamete type, a second gamete type, both of these are viable. I encourage you to practice with these. There are a couple in your lectures as well, and Pierce has some really good examples. Well, this is another chromosome aberration problem. It says that an individual has the 4 chromosomes shown there where the periods represent the centromeres. This time there really are 4 chromosomes. And you want to find out what kind of chromosome aberration you have. Well, let's see. If I look at these first 2, they both start with BC, but then they change. If I look at these 2, there's the HLM, the GHLM, but it had XZ, that has XZ. And if I look around the centromere, one centromere is between G and H, here and here, and the other is between S and R. So these two are homologous centromeres, and I'm just going to make that centromere an open circle. And then these centromeres, I'll make a solid end circle. The two solid ones are homologous, and the two open ones are homologous. It's always important to figure out which centromeres are homologous because those have to synapse. And it appears that one of these has BCF, one has BCT. So there's a break there. And the ZX also appears to be the part that's different. See, FG centromere, HLM on the solid centromere, and either ZX or BC. Similarly, TS centromere, RQ, and either BC or ZX. So we have a reciprocal translocation where the BC was moved from one chromosome and the XZ was moved from another, and they switch places. So reciprocal translocation. So reciprocal translocations synapse in a cross-type structure. So we can draw that outline. And again, synapsis occurs when there are two chromatids on every centromere. So these are the 4 chromosomes. Here is the second chromatid on each. And we'll put the centromeres out in a little bit where we figure out where we want them. That's one line. Okay, you can start labeling with any of these. And I usually just pick whichever one was listed first and start putting it on the upper left, but it doesn't matter as long as you end up pairing the homologous centromeres and pairing the homologous genes. So BC, and the line I've drawn is the corner, these interior corners on my cross. So I'll have BC, and then, a lot of stuff here, FG centromere, HLM. All going up this way, F, G, and this is the closed centromere, H, L, M. By the way, when you take your exam, you get to use blank scrap paper. You pick it up from the Delta Center or whoever your remote proctor is. And I encourage you to take several pages because Exam 2 has drawings like this you might have to draw out, and it also has lots of mathematical calculations, so you might want several pages so that you'll have room to draw freely. Okay, so my other BC goes to the breakpoint. There's my interior corner, TS, open centromere, RQ. Now remember, across from Q has to be the other Q. So across from Q, this is the chromosome we haven't done yet, Q, R, open centromere, ST, breakpoint, X, Z. And this last one, Z, X, breakpoint, F, G, closed centromere, HLM. So we've got them synapsed correctly. Now gamete formation. When the gametes are formed, every gamete has to have a closed centromere and an open centromere. And that's just based on Mendel's law of independent assortment. So we would not have these two going to the same pole. That would be non-disjunction. We won't have those two going to the same pole. They both have the open centromere. That would be non-disjunction. But we could have the two on the left going to one pole and the two on the right going to another. Closed, open. Closed, open. So let's draw what that would be. Now think about meiosis, chromosome synapse. I can't make my fingers do the cross. In the first meiotic division, the homologous chromosomes separate. In the second, the centromeres split. So when I'm drawing the gametes, I'll only have one chromatid on every centromere, even though I have two right now. So one of these gametes could have the chromosome on the left, B, C, F, G, closed centromere, H, L, M. And this other one on the left, B, C, T, S, open centromere, R, Q. Now look at these. They both have BC and they don't have XC. So this is not viable because it has duplications and it's also got the deletion of XC. If I had this going toward one pole, the stuff on the right would go toward the other pole. So let's draw those out. M, L, H, G, F. Oops, forgot the centromere. It's between H and G. It's a closed centromere. X, Z. There we go. And the other one is Q, R, open centromere. S, T, X, Z. And again, two copies of XC, and it's missing the BC. So this is not viable. When we separate this way, this is called an adjacent segregation pattern. The other segregation pattern is called an alternate segregation pattern, but basically it's diagonally opposite chromosomes go to the same pole. So top left, the bottom right would go one way. The top right and bottom left will go the other. So let's do top left, M, L, H, centromere, G, F, C, B. Bottom right, Q, R, open centromere, S, T, X, Z. And then the last one is top right, that was a closed centromere, and bottom left. And those two are viable. One more problem. In the lectures, we talked quite a bit about the production of amphidiploids. These are in plants because we can mess with chromosomes and double chromosomes in plants and they survive. It's harder to do that in animals. But with the amphidiploids, we're looking at mating two species together, getting a sterile F1, doubling the chromosomes with colchicine to get a fertile plant. We looked at some of our crop species like wheat, which is actually a combination of 3 species going together, and cotton would be another. Peanuts, there's several others that are following this amphidiploid pathway. So here we have 2 species. Species A is 2n equals 40. Species B is 2n equals 18. Something that's really important is that these species are not related to each other. And that means when we cross them together, that F1 is sterile and those chromosomes do not pair up. So all we see in meiosis are univalents, chromosomes that are not paired, versus bivalents would be when 2 chromosomes synapse. So we cross species A and B together. Let's do that. Species A is 2n equal 40. And species B, 2n equals 18. The gamete from species A has half of the 40 or 20 chromosomes. The gamete from species B has half of 18 or 9 chromosomes. And they said that when we did that, we formed hybrid X. Hybrid X then must have 29 chromosomes. We can go ahead and fill that chart out. 29 chromosomes. None of these chromosomes are going to be pairing up. So we have 29 univalents and 0 bivalents in meiosis. Then it says we treated hybrid X with colchicine to make hybrid Y. Let's see. Give myself a little bit more room to write. Hybrid Y must have 2 times 29 chromosomes, so that's 58 chromosomes. Remember I said colchicine doubles the chromosome number? It does that because it disrupts the spindle fibers. So where the chromosomes normally will line up in mitosis and then split, these spindle fibers are disrupted, and so the chromosomes just all hang out, and then they all end up getting incorporated together, and we end up with twice as many chromosomes. So hybrid Y has 58 chromosomes. Now, the cool thing here is since we doubled each of these chromosomes, there really are 2 copies of each of these 29 chromosomes. So there are no univalents and 29 bivalents in meiosis. And now let's see, what do? We have a hybrid Z. We made hybrid Y, that's this one, to species B. Okay. And we can think of this hybrid Y as 2n equals 58. Because it's going to behave like a diploid. It's an amphidiploid. So to get, let's see, this is going to be hybrid C. Species B has 18 chromosomes, so 9 go into the gamete. And hybrid Y has 58 chromosomes, 29 go into the gamete. 29 plus 9, 38 chromosomes. Put that in our chart too. Now, this is kind of interesting. Remember up here, species A and B were not related to each other. However, hybrid Y and species B are related to each other. In fact, 9 of the chromosomes in this gamete are the same as the 9 there. So these chromosomes will pair up to make 9 bivalents in meiosis. And that means there are 20 chromosomes that aren't pairing up, and they are the univalents. Now that one is a little tricky to wrap your head around, I do understand. So please look at it carefully and let me know if you have any questions. There's one more part to this problem set, and I'm not going to go over this because you can read it. You don't need me to read it to you. However, this is actually a really good chart for some of the qualitative information. It does not tell you about all of the different aberration types, so you will want to go back and get some of that. But it does tell you about some of these. And so you have some of the vocabulary that might be important, things like a terminal versus an interstitial deletion. That would be important. Remember, an acentric fragment is the fragment that's lost. I'm not sure if that word is in here. That's another word that is important. when that is the name of the part that is lost in the deletion. And it tells you some ways that this can originate, and then it tells you how it can affect that individual that has it when it originated, and how it can affect the offspring. So there are probably a whole bunch of qualitative types of questions I can come up with based upon this page. So I encourage you to read it and feel very comfortable with explaining how these different types of aberrations arose, the definitions of the various terms that we have, and the effects of the individuals and their offspring. Oops. Yep. Well, thank you very much for your attention. I hope that you understand this a little bit better now, and I hope you have a fantastic day.